So, Dr. Varghese, the talk is Are Animals Still Needed in Research? Exploring 3 Rs: Replace, Reduce, Refine, and 4 is Funding.
Right, thank you very much. So my talk is much more generic. Probably it's not tailored to a pancreas crowd, but just looking at this broad question of whether animals are still needed in research, and if so, what are some concerns and how we can address some of those. So over the next 15 minutes or so, I'll go through briefly the 3 Rs, which is the bedrock of all animal ethics related to animal experimental research. So the replace, reduce and refine principles, and following which we'll have a short discussion on problems with research based on animal models. So this is basically the bedrock of all animal ethics. So whenever we look at animal-based research, whatever the question is, so these are 3 things that we need to pay attention to: replace, reduce, and refine. And this framework was proposed in 1959 by Russell and Bruch. And basically, they asked 3 questions. So one is replace. The question here is, do you really need animals to answer the research question? So it is related to ways in which you can avoid the use of animals. So how can you avoid the use of animals? One, you can do a full replacement. For example, if the research question can be answered by in vitro models, cell culture models, you should go ahead and use it. Or in silico models with computational models, can you answer the question appropriately and to the scientist's satisfaction without actually using animals? So in vitro and in silico replacement. Or you could do a partial replacement, which means that you replace a sentient animal with what we consider as less sentient. Sentient basically means is the capability to experience pain and suffering. So if you can replace sentient animals with organisms which probably are considered to be non-sentient, like the flies, the Drosophila and nematode worms, then you should look at a partial replacement as well. So that's the first question. Do you really need animals for your research? So if you can answer that as yes, animals are indispensable, then you actually go ahead and look at the next principle of reduce. So what does reduce mean? So in the past, when animal ethics committees looked at this particular principle of reduce, it was that you just cut down the number of animals. So if the experimenter would ask for, say, 10 animals, you just give 5. So researchers would work around it by, if you really want 10 animals, then you ask for 20, because you know that the committee will just reduce it by half. But that is not, what this principle basically means. So the principle of reduction is to use the minimum number of animals that is required to reliably answer your question. So that is the principle of reduce. So if you have a question that basically needs 50 animals, then you should go ahead and use those 50 animals. But that must be justified with a proper sample size, right? And the concept of doing a sample size calculation for animal research is very poorly developed, because most people go with standard stereotypical numbers like n equals 6 or whatever, or n equals 3. There isn't really a strong rationale for using these numbers. But the principles of calculating a sample size for an animal study is the same as the principles that you would do for a human study. So in a human study, calculating a sample size is absolutely essential, right? And everybody does it. But when it comes to animal studies, for some reason, the importance of doing a proper sample size calculation for an animal study hasn't just gotten on. So the principle of reduction basically means that you put in the time and the effort that is required to search the literature, maybe do preliminary experiments, and on the basis of that, use a proper statistical software to calculate a sample size. And once that sample size is calculated, is justified, you present it to the Animal Ethics Committee. And I'm happy to report that in most cases now people are enlightened. Most Animal Ethics Committees will actually approve the number of animals that you have asked for if you have properly justified it using scientifically justifiable sample size calculation. Right? The other thing with animal experiments is that the good thing about it is that when you use an inbred strain or an animal model, you expect the background genetic variability to be less, right? If not minimal, which means that the background variability that you often see with human studies is not what you would see in animal studies. They're more of a homogeneous population. But on the other hand, the variability is actually introduced by the procedures that we do. So if you are not going to standardize your procedures well, and if your skills at a particular procedure is poor, you introduce a lot of variability, right? And when we all know that when the variability is high, then you need larger number of animals to get a statistically significant answer, right? So as researchers, what do we need to do? We need to properly design our experiments. It's really important. Calculate a solid sample size and make sure that our experimental variability, the variability that we introduce through the experiments that we do, our procedures, need to be kept at the minimum. And when we do this, we will be able to apply the second principle of reduction or reduce. So, at this stage now, you have answered the question of do I need to use animals? Yes, right? And then the second question would be, how many are you going to use? Yes, I've calculated a sample size and this is the minimum number that I need. And the third principle is refine. So how are you going to ensure that the animals that you're going to use are treated as humanely as possible? You take care of their well-being, you make sure that pain and suffering is minimized as much as possible. And that is what is addressed with refine. So in refine, we actually make sure that if possible, avoid painful procedures. If not possible, use analgesics and anesthetic agents at appropriate doses, and probably set humane endpoints. So this again is very very research question specific. So you have to apply your mind to identify these humane endpoints, to make sure that the animals are getting analgesics at sufficient doses. So all of this comes out of an ethical obligation to make sure that animal ethics and animal suffering is minimized as much as possible. So that's basically the bedrock of animal ethics, the 3 R's. So one is replace, if you don't really need to use animals. Reduce, use the minimal number of animals that you need to reliably answer your question. And 3, refine, and make the suffering of animals that you induce as minimal as possible. Right. So that is basically the point of view from the point of view of animal research ethics. But there's also another point of view, the scientific point of view, right? And that's basically the question of, are animals still needed in research? And I think Dr. Ashok Saluja's talk kind of alluded to the fact that there is a fundamental role for preclinical models, animal research, in looking at, sometimes finding pathogenic mechanisms, in other times looking at potential therapeutic agents, etc. So the question is actually rhetorical, because I think all of us in this hall can agree that a lot of fundamental discoveries across history have happened first by, through experiments done on animals. We look at the discovery of insulin or vaccines, the list is very long. So the question of, are animals still needed in research? Like I said, the answer is a resounding yes. But what is the problem here? There are a number of problems with the way we actually do these experiments. One is translatability, right? And so the question of how sure are we that the findings that we get using an animal model can be translated to humans? And that is a fundamental question. Mice are different from human beings. And even though you can argue that 95–96% of genes are the same, we all know that they are fundamentally different. And many things that that work in animals probably don't end up working in human beings. So there is a problem with translatability which we have to live with. The opposite is also true. I mean, in many occasions, something that's underappreciated is the fact that sometimes things that don't work in animals probably do work in humans. So that is actually a consideration that we often don't, we don't really think about it. But both are true. It is true that many things that work in animals may not translate into human beings. Probably also true that many things that don't work in animals might be useful in humans. So that's the question of translatability. Very little that we can do about that. But then there is the question of reproducibility, and I'd like to talk about that a little more with the time that I have left. And last is reporting. So reproducibility. So these are a couple of papers, slightly old now. So this is these are studies that have come out of the pharma industry, and they basically were looking at validating the results of landmark studies published in journals like Nature and Science, etc. And this one is basically similar, and they looked at cancer. So what both these papers are showing is that, look at this one, only 14 out of 67 studies, 21%, 1 in 5 studies could actually be replicated, right? So we're not talking about translatability here. All they are trying to do is to reproduce the findings that have been published, right? And that is a pretty low number. Only 1 in 5 studies were reproducible in terms of their primary outcomes. And it comes to this, it's even worse here. So 6 out of 63, about 10% of preclinical cancer research studies were reproducible. So I think there is something really wrong here, right? So why is it that some experiments only work in some labs? Right? There has to be an answer to that question, because billions of dollars, billions of rupees is spent on these preclinical studies. And if they cannot be reproduced, I think there's a fundamental problem here. And what could be those problems? There are many problems that could be there. One of those problems is the reporting, right? So here again, a study which showed that, among published animal research, about 12% of publications actually reported on things like randomization and blinding, which are very fundamental to human studies but completely neglected. Sample size justification absolutely missing in all 100 studies that they considered. Poor description of statistical methods here, and in many cases the reporting was non-standardized, and because people don't report these studies well, it's probably the reason why it can't be reproduced as well. So there have been attempts to ameliorate this problem, and like the consort guidelines, we have the ARRIVE guidelines, which basically means animal research reporting of in vivo experiments. So this is basically a checklist that encourages researchers to make sure that some of these fundamental things are actually alluded to in their paper. It's a checklist. So it's kind of a reminder to a person, a scientist writing a manuscript, to ask himself or herself whether they have reported all these critical things. Checklist. So it just tries to make sure that the important aspects of methodology are not missed out. And when it's reported, then an independent third party can probably reproduce those results, if indeed they are true. So many journals now actually require you to submit the ARRIVE guidelines, the ARRIVE checklist, along with annual studies. There have also been attempts to improve experimental design. For example, you have the NC3R, that's the National Commission for Implementation of the 3 Rs, have provided tools like the Experimental Design Assistant. There are tools for sample size calculation. You can even prospectively register your annual study, right? Again, something that is mandatory for human trials. You can actually do that for annual studies as well. It is not legally mandated, but highly recommended. So this is just one of the sites where you can prospectively register your annual study. So these are things that we should take seriously. People in this field have to take seriously and make sure that we follow best practices. So what is my take-home message? The 3 Rs basically is, just tells us that it doesn't matter what we think or whether we love animals or not. It is an ethical obligation for all researchers to make sure that animal welfare is taken care of. It's an ethical obligation. And from the scientific point of view, we need to remember that good animal welfare produces reliable and reproducible results, which basically is good science. So animal welfare needs to be taken care of from both the animal ethics viewpoint and also from the viewpoint of producing good science. So that's basically my take-home message. And to summarize, when we do animal research, let's take the 3 R's seriously, and it's important to know that experimental design and reporting of animal research are very important, and we need to pay particular attention to that. And that will go at least some way in addressing the skepticism that many people have with regard to experiments using animals. So these are some of my references, and I want to thank you for your time and attention.
Great talk, Dr. Bunyan. Congratulations. And it's coming from someone who has probably used 100,000 mice in my lifetime. So I think you gave a very succinct importance that why one needs to be right. Animal welfare obviously is important for ethical reasons, but to me is more important for your results, for your science. And doing appropriate experiments with the right numbers and right experiment is very important. Actually, when I look at the literature, including many of my own research publications and experiments I have done, I think we could improve on most of them. When we say only like 10 of these are true without 100, well, these are not that someone intentionally made mistakes, but the development of the field was not up to the mark. I'll give you just one example, and I'd like to see what you think is that. So when we studied, let's say I study a lot of pancreatitis and pancreatic cancer, and I'll give you a couple of examples in my next talk. But when we study pancreatic cancer, we use mice, which are 6 weeks, 8 weeks, 10 weeks, most of the time, even for pancreatitis. This is a pancreas conference. But it's true for all diseases. Even cardiologists will use mice which are 8, 10 weeks. And in human age, that is 2 years, 5 years of, 6 weeks of mice.
So in general, about 8 weeks would be considered adult. So maybe it's around
but
18 years in humans.
The question is that, now compare that with humans. A typical person who gets a cardiac disease or person who gets pancreatic cancer is 65 years old. Pancreatitis is 40-some year old. We are studying a process in 8 weeks old mice, and then we are going to use that to apply. So I think we still need a lot of refinements in this area. I haven't seen one study in pancreatic cancer where they use equivalent of 60-year-old human in doing that. It's failure of my own research, but it's true for everyone. So I think your talk is very, very important, that we need to be very careful in planning studies. Thank you for doing a great job.
Yeah, just a response, I couldn't agree more. And I think the rigor that we now use for human studies, in terms of sample size calculation, human ethics, and things like that, I think we had to bring the same kind of rigor to animal science as well. And that's where I think the field is lagging. I think we as scientists would be the best persons to address that gap. Yeah, thank you very much.
So it's a question, you are familiar with both the spectrum, animal ethics, and you're an active animal researcher. Where should we start to negate the problems you highlighted?
Yeah, so one thing is that there are no external agencies, Chris, that actually mandate any of these things. Like for example, there is a really useful tool called as Experimental Design Assistant, available free of cost from the NC3R. And agreed, it's not very user-friendly, but it's actually an excellent tool, especially for novice researchers, to start off with designing an experiment. For example, simple things like randomization. Do you give any thought about randomizing or blinding animal studies? We don't. We just unleash our students on the mice, so to speak. And we just tell them that take 6 rats or 6 mice, divide it into 2 groups, and you give this drug to this group. But there is actually a place for all of this. I think you can randomize, you can blind. Like for example, you don't have to tell the student that this is the test drug and this is saline. You can just name it A and B. It is extra work, but I think it's work that is well worth spending the time on. So to just answer your question, I think the buck stops with us as researchers in the sense that when we design our studies, we need to make sure that we pay attention to these small things which are not legally mandated or enforced.